Good morning. I'm Kevin Crilly, Executive Director of Gift Planning. Welcome to the 31st Annual Wheeler Lecture and now Luncheon in this, the 150th year of the University of California. So go Bears. So on behalf of the Office of Gift Planning and Chancellor Christ, who will speak to you briefly at lunch, we want to welcome you. But I want to get directly to our featured speaker this morning, because I think she has a lot of fascinating things to say. And for those of you who might be intimidated by science, like me, I know she's going to make it accessible. Professor Amy Herr is the Lester John and Lynn Dewar Lloyd Distinguished Professor of Bioengineering at UC Berkeley. She became a full professor in 2015. She has a BS from Caltech and a master's and PhD from Stanford. Prior to coming to Berkeley, she was at Sandia National Laboratories. Her CV is very long. She's received numerous awards. The few that I just wanted to point out, because they always appeal to me. In 2007, she received the Outstanding Mentor Award from the Sandia National Labs, and in 2012, the Outstanding Instructor Award in Bioengineering here at Berkeley. So without further ado, Professor Amy Herr.
Good morning, everyone. It's wonderful to be here. Thank you so much for taking time from your day to spend a little bit of time with us. My name is Amy Herr. I'm a professor of bioengineering. I've been at UC Berkeley about 11 years now, and it is a tremendous place to be. So I'm happy to be able to share this campus with you. Today, I've been asked to tell you a little bit about the research that goes on in my group, and I'm going to do that in a way that hopefully is interesting to you. And I'm also going to stress some of the impacts that come from that research, both in terms of the new knowledge and ideas and capabilities that we work very hard to contribute, but also in terms of the people that we train here and what they go on to do in their careers. So if we start off with the research, if you look around the room, you'll notice that we all look very different. If we dig inside and look at our DNA or our genome, it turns out that we're all pretty much the same. Something else that we unfortunately have in common is that we all also get sick. That may manifest in different ways for different individuals, but it is part of being human. If you take a look at what's happening inside the body before we start manifesting symptoms, there are changes going on, changes happening at the cellular and the molecular level. If disease that we've acquired is something that is very serious, that may make changes to our genome or our DNA. Other disease states, as we know, can be fleeting, something that's just very temporary. So this is what my research group works on. I did mention that I'm in bioengineering, so I am an engineer by training. We work to design and develop tools that can help us to understand the changes that happen during disease development, during disease progression, and also how our body responds to therapy. So that's why an engineer is working in the life sciences and in medicine. So when we look at humans, when we look at individuals, you probably all know that we have differences. You probably all know that the differences also manifest in how our disease progresses. This is true even down to the cellular level. So if you look at something like a solid tumor in cancer, we understand that there is tremendous variation among the cells that even make up that small mass. In addition to that, if we have other disease sites, there will be a lot of variation between how the disease manifests in those different sites as well. So this is part of the heterogeneity of life and disease that my group works to measure and to understand at the cellular and the molecular level. So one big component of what we do are to try to identify what we call biomarkers, or signatures of disease. So these are biomolecules that we can measure as part of diagnostics, and that gives us a readout in terms of the current state of disease or response to therapy. So the challenge with biomarkers is actually a pretty big one. We know from the Human Genome Project, so a schematic of DNA at the upper left of the screen here, we know from the Human Genome Project that a lot of information is locked inside of DNA, and we're starting to access that through genomics and moving genomics into personalized medicine. DNA, however, is somewhat static. It's not entirely fixed, but it is not a dynamic biological molecule when compared to the downstream product of DNA, which is protein expression. So if you think of DNA as maybe the blueprint of life, which is something that it's often referred to, your genome is the blueprint of life, you might think of these downstream proteins as molecular machines. They're dynamic, they're changing all the time in response to the environment that is built by DNA. So you might think of them as the mopeds or the vans or the cars that zip around the city that is built by DNA. In some ways, from the biomarker point of view, this is really intriguing to us because there are hypotheses that proteins may be more reflective of the current state of disease, the local state of disease, than perhaps this more static DNA is. And that's a tremendous opportunity when we think about how in real time can we understand disease, how can we classify disease in real time, and how can we act to treat disease effectively in real time. That's one of the bonuses, I guess one of the promises, if you will, of proteins as biomarkers. There's another side of the coin, and that is the challenge of proteins as biomarkers. So proteins, as I mentioned before, are dynamic. They are always changing. These molecules are continually being expressed and secreted. They're continually being modified by chemicals, decorated with small chemical moieties, changed, cleaved, binding, interacting with each other. And that, again, is a tremendous opportunity because it may give us a window into how a disease is changing. But the flip side of that coin is that the measurement and identification of which proteins are the best biomarkers for a specific diagnostic question is really a monumental challenge. It is not hyperbole to say that there are literally millions of different forms of these biomolecules in our bodies right now. And so my research group works really hard to essentially develop what I'll call a stethoscope for cells, to be able to measure and then understand, make sense of what do those protein signatures mean and how can we use them to reflect and understand real-time disease. And so we oftentimes look at proteins directly to be able to bring together that understanding. And we do it at the cellular level. So we look at solid tumors, we dissociate those tumors, and cell by cell, we're interested in understanding what's happening inside of each of those cells at the level of proteins. So this stethoscope for the cell is really what we're working on. So this, as I mentioned before, is a pretty big challenge. Maybe I should actually say it's quite a small challenge. So a human cell, a mammalian cell, is about 1/1000th the volume of a human teardrop. So each of these cells is just a minuscule amount of material and very small in size as well. You're looking here at just a scientific, artistic depiction of the head of a pin with a red blood cell sitting on the head of the pin, just to give you a sense for the sort of scale that we're talking about when we're talking about peering into each of individual packets of life, these cells that we're interested in profiling to understand disease. So the way that my research group chooses to do this is a little bit different. We essentially make these tiny cups, and you're looking at a confocal micrograph, so just a micrograph image in 3D, a microscope image in 3D of one of the tiny little cups that we make. The cup itself is made in something that's known as a hydrogel. It's pretty much like Jell-O, so it is a very aqueous type gel material. It's got a lot of water in it. You're looking here in that micrograph of 2 mammalian cells that are nestled in one of those wells. So you can see that we're able to isolate and encapsulate individual cells using these small cup features. I'll tell you in a minute what we do with these microstructures in order to measure and analyze the protein contents of the cells. But what I want to tell you first is how we make these tiny structures. And it's actually thanks to something that you probably brought with you today. So in your pocket or maybe in your pocketbook, you may have brought your cell phone, you may have a laptop with you or a tablet with you. So the very same structures that are used inside of your electronics to route electrons and subsequently information to you are the same types of fabrication approaches that we in bioengineering have adopted from the electrical engineers, adapted so that we no longer route electrons necessarily, but we route cells and biological molecules. So the fabrication processes that were developed to power the electronic chips in all of the devices we use are now being used to understand and measure molecular level signatures of disease. And for us, a big part of that are the amazing facilities that UC Berkeley has available for researchers to make these types of microstructures. So you're looking here at an image of one of Berkeley's most outstanding resources, and I would say probably the best semiconductor cleanroom facility in an academic setting in the world. Berkeley really is the birthplace of many of these fabrication technologies and approaches. And so we work in facilities like this to make these tiny structures to be able to manipulate and handle individual cells. I just wanted to show you an image of what some of our structures look like on devices. So this is the type of chip that we make, maybe a little bit different than the semiconductor chips that you brought with you today. But essentially, it's just a glass microscope slide. You're looking at sort of an end-on view here. The edge of the glass microscope slide is kind of to the bottom right. And then on top of that glass microscope slide is a thin layer of this hydrogel material. I hope that you might be able to see the microwells that are stippled in this material, this hydrogel or jello-like material that's sitting on top of the slide. If you can't, I've drawn a little tiny circle there to pull out that micrograph that I showed you earlier to show you how we isolate the cells using a very simple device like this. One of the main aspects of my interest is measurement science, but I will be honest and tell you I also have quite an interest in chocolates as well. And so I love the opportunity to bring those two interests of mine together. But as tied to what I'm telling you about today with our ability to peer into a cell and to understand what's happening, as we move forward with personalized medicine, we've come to realize that some of the approaches that we use to, for example, classify different types of cancer really only look at the proteins that are on the surface of a cell. So many of our approaches don't peer inside the cell to understand what's happening with the proteins on the inside. And interestingly enough, if you like chocolate, you may understand this, the outside of a cell, much like the outside of an assortment of chocolates, can look very much the same from piece to piece, if you will, or cell to cell, if you will. It's when you look in the inside, there are big differences. And if you're someone who maybe doesn't like coconut so much, you might want to know what's in the inside before you take a big bite of that piece of chocolate. And so for us, we're very interested at the molecular level to look at the inside to understand which cells are changing their logic in a way that we don't want them to change it early. Which cells are some of the early cells that are becoming more and more diseased as compared to the surrounding cells, say in a solid tumor? So for us, the tools that we're developing are to peer inside of the cells to look at what's happening. And as I mentioned, on the right-hand side, that's a semiconductor chip. Again, you brought something similar with you probably today. And below it is the logic that's used to route those zeros and ones is a conduit for information and the logic. And on the left-hand side is sort of the parallel, what happens inside of a cell. So instead of showing these different unit operations for a semiconductor chip, on the left side, each of those little ovals represents a protein and how the proteins are part of a larger overall signaling network. That's how the logic inside of a cell takes place. I'll leave it at that for today, but that's the parallel there for sure. And so for us, we want, much like this egg that I show here, we want to break open these tiny, tiny packets of life, these cells, 1/1000th the size of a human teardrop, and we want to look inside. And this is where physics is not really our friend. And this is why we have to use some of those advanced fabrication approaches that I talked about earlier. So with a tiny, tiny packet of information like the cell, when I break it open, it's in an aqueous environment, it's in fluid. When I break it open, diffusion very, very quickly acts to dilute out the contents of the cell. Much like you might imagine dropping a packet of dye or ink into a glass of water, or maybe even into a swimming pool. Very, very quickly, you're not going to be able to see that packet of dye that you've dropped into the pool. That's because diffusion is diluting out that packet of information, that packet of dye. So for us, when we break open a cell, we have to act very, very quickly, or all of those molecules that we want to measure will be diluted out into the surrounding fluid, and we can't make measurements when that dilution is too great and the cells are in too low of a concentration. So for us, what we do is we essentially take those tiny, tiny cups that I showed you earlier. So in the top, you're looking down on one of those cups. You can see that circle, and maybe in the back, you can see there's a green, false-colored cell that's sitting in that cup. We break that cell open in the cup and very, very quickly apply an electric field and drive all of the molecular contents of those cells from that cup to the right in the surrounding hydrogel material. So this allows us to lock in place. We covalently link our proteins to the gel so that dilution by diffusion does not hurt us in terms of being able to measure the proteins that are there. In addition to that, you're going to notice that there are now two spots to the right of that microwell. That's because we've separated two proteins of interest that we couldn't detect when they were both together inside the cell. So we've spatially separated them so we can measure each one of those proteins of interest. In some cases, the differences in the chemical composition of those proteins will be quite subtle. And so it's important for us to think about how we do that separation process, and that's something that my group works on extensively. We do this, of course, for more than one cell, so I just wanted to give you a glimpse of what a tiny array of 400 or so individual cells look like. So each of those little blue stripes starts in a microwell, one of these little cups that holds the cell, and the information about the cellular protein contents is encoded within that colorful streak from the left to the right of each of those microwells. That's how we profile the protein signature of each of the cells of interest. All of this really goes to our bigger vision, which is to help scientists, life scientists, and to help clinicians develop essentially a network map, if you will, of disease in each individual. And I hesitate because I use the word map. I think it's more something like an air traffic controller or traffic, sort of real-time traffic reporting. We're interested in developing these tools so that they can be used routinely to understand how your disease is developing or responding to specific therapies. And that really leads to the larger vision of precision medicine overall. So when I say we in my research group, there's an amazing group of individuals who work with me every day, have the pleasure and the honor to train and to collaborate with. This is just an image of my current research group. We're up in Stanley Hall here on campus, an amazing, again, resource for the entire campus there with core facilities and world-class labs. Most of these are graduate students. Most of these individuals are graduate students, and almost all, if not all of them, in this photo have received either federal research grants or named fellowships, which allows them to pursue their graduate research. So one, as you probably know, that helps them to train to be informed leaders of the future in this area of bioengineering. But it also helps us to conduct the research that we're conducting, in addition to federal foundation and industry grants that I put together, I apply for my research group. So I just put up a couple of aspects of Berkeley's standing in the world, if you will. You probably are familiar with most of these. Obviously, we're considered number 4, top 5 global university, number 1 public university. I also want to mention that we are the number 1 choice for graduate students who receive the prestigious National Science Foundation Graduate Research Fellowship. So that fellowship is quite competitive. And it is also life-changing. I myself was fortunate enough to be awarded one many years ago and to benefit from the investment of our government in the next generation of scientific leaders and thinkers. I think one big thing I want to point out here is it is because of our standing in the world and the research that goes on that these individuals choose to come to Berkeley. They compete to come to Berkeley for their graduate studies. And what you are doing to help support Berkeley is helping to maintain that for the future. It is a compounding effect, as I've seen over the past decade or so. what happens when some of these leaders decide to go on and create their own future? So when they graduate, I just wanted to give you two sort of brief snapshots of some of our alumni. So Dr. Kelly Gardner was a PhD student with me. I think maybe she was my 5th student. She also had an NSF Graduate Research Fellowship. When she finished, after she defended her thesis and submitted her dissertation and published her papers, she was, not surprisingly, sought after by many industry positions and some prestigious fellowships, one being the Kauffman Foundation Fellowship, which would have allowed her to go into venture capital right out of graduate school. So she accepted that and started moving forward and ended up stopping by my office one day as her advisor. I definitely have that door open to her, as I do all the students on this campus. And she said to me, Amy, I'm really interested in doing something a little different. And of course, I kept calm because that's quite a prestigious fellowship that she had been awarded, and it was quite a competition to get it. And I said, oh, what is that? What are you thinking? And she said, I'm thinking I really want to start a company. And I thought, okay, we're kind of in the epicenter of entrepreneurship. That makes sense to me. Do it. I encouraged her as much as I could. To my surprise, she looked right at me and she said, and I want to do it with you. At that point, I'd already given too much support, so I said, I gotta go with this. So I said, let's do that. So we ended up getting a couple of other members of our team. She was quite interested in the technology that I just described to you. These tiny cups and this simple chip that was developed and invented by myself and another graduate student who's gone on to an amazing start to his career in academia. She was interested in licensing that intellectual property from UC and moving forward with commercialization. So she did that. She ended up taking everything that had come out of the federal foundation and industry support for our work, these fellowships that powered the student involvement, graduate student involvement. She ended up taking what is, for me, just a highlight to see this sort of experimental setup, but which is quite embarrassing when you think about what a product would look like. She took these systems and she basically, in 2.5 years, brought that prototype system into something that would be a product that she could sell as CEO of the spin-out of UC Berkeley. In that process, she used every resource available to her in UC and on the UC Berkeley campus. And when I say every resource, I mean every, and there are a lot. I don't know how often you are able to spend time on campus, but this campus is, as I mentioned before, the epicenter, not just for scholarship and teaching, but for entrepreneurship, which is impact, direct impact on our society from what's happening in our research labs and in the auditoriums and lecture halls like this. So she took that product out. It ended up that some of the existing large biotech companies were so intrigued with it that one of the large companies in the US acquired that startup in 2016. The product that's been developed out of that is now sold worldwide, and in addition to that has received industry accolades for its performance, which is, as a faculty member, something that's pretty amazing to see. Something that was invented in your lab, and we spent many years and many dollars and many blood, sweat, and tears, I'll just say it, working over, actually out in the world being used in such a short time frame. So that's what can happen from projects that become products when we start thinking about how do we have impact on the world around us. That is certainly one way. But there's another aspect to me as a faculty member that's really, really important to point out, and actually, quite honestly, much more important. And that is what I've seen in terms of the environment that Berkeley creates for launching careers. So the next generation of scientific leaders, business thinkers, contributors to our world, I think are summed up in someone like Dr. Gardner. So she was recognized soon after the acquisition of that tiny startup by MIT's Technology Review magazine. It is a respected technology journal, and she was recognized as one of the top 35 under 35 years old in that year. It's interesting to me, she was not recognized for the research that happened in my research group. She was recognized because she was able to figure out how to take what are called lean or agile methods that are commonly used in the software industry and apply those to a completely different industry, the life sciences tools industry. I don't know if anyone here works in life sciences tools, but it is a very slow-moving, high capital-intensive industry, very, very different than the industries that are used to make software and apps as well. So that for me was a pretty important insight to realize that some of these efforts to spin out technologies also result in spinning out amazing new business leaders as well. This isn't just Kelly and an example of her path, but also actually my first graduate student, Dr. Kwesi Poori, is also spinning out a company based on UC's intellectual property and just finished Y Combinator and is moving forward with his startup as well. And that's his photo, the small photo at the top. So I just wanted to give you a sense of that. I'll also tell you that this whole experience really crystallized for me, beyond my previous understanding, the scope of the importance of investment in the next generation of, one, products and solutions to solve big problems, but also the next generation of leaders. And so I've devoted actually quite a bit of my time to meeting with and working with government relations here to talk to policymakers, both at the state level and at the federal level, about the importance of the investment of education, public education, and STEM, science, technology, engineering, and math, as well as programs that help with commercialization of inventions and discoveries from our labs and support through fellowships of some of the young budding leaders of tomorrow. So I do that through my own work with the government relations here, but I also do that as a faculty advisor to the Society of Women Engineers. It's about 400 undergraduate women and men who are engineers who most recently spent 3 days on Capitol Hill talking to policymakers about the importance of us investing in their future. So it's quite an honor to be part of spreading that message as well. Another message that I just want to spread a little bit here in this part of my comments is maybe some aspects of Berkeley that you might not be so familiar with. So when I talk to people I know, a lot of people understand that we're the #1 public, and I always say, aha, but top 5 global, which is a bigger pool for sure. Not competitive at all, that's for sure. But there's some other things that I've come to find out as I've had the opportunity to be involved. Being here at Berkeley, I try to think forward-looking, but really the honest truth is it really is the students who end up pulling us forward, and I just try to basically facilitate their efforts and get out of their way. And a big part of that is in this area of making a positive impact through the work they do and the careers they choose. So just 3 short things here. Number 1, if you're not, or I should say number 3, if you're not familiar, UC Berkeley is considered the #3 university for venture-backed startups that are led by women, which is an amazing rank to have globally. In addition to that, Forbes, in their most recent rankings, named UC Berkeley the #3 startup university. So definitely seeing something there in terms of the impact that we're having from the work that we do on campus. Number 2, for venture cap startups, regardless of the gender of the founder or the CEO of the startup. And then number 1, PitchBook last year, or this year actually, named us as the #1 university for students who are thinking about becoming entrepreneurs. So I just wanted to share this with you because I oftentimes find that people may not be aware of how much of a powerhouse Berkeley is in terms of the state of California's economy and our global economy as well. And then, importantly, as an advisor, the people that we're training by these experiences that are being provided on this campus. So Berkeley is leading the way. I oftentimes spend time traveling to other universities to present research talks, so not exactly what I'm talking about today, but touching on a little bit of what I've shared with you today. In those meetings and in those visits all over the world, I have a chance to meet with colleagues and compare notes. One, compare notes about what they're researching, how they're approaching problems and what they're thinking about, also how they're training students. But I also have a chance to kind of see what's going on at these different universities. UC Berkeley in 2012 started just what I will classify as a pioneering program to support the education and efforts of faculty who think they may have inventions or discoveries that might someday have commercial impact. So the Baker Fellows Program was started in 2012 by the vision of an amazing supporter of this campus. I was so fortunate to be named to the first class of Baker Fellows, and I can say without that fellowship, the story that I've told you about Kelly and our startup, Zephyrus is the name, would not have happened at all. So I've yet to find another university that supports faculty, potential faculty founders, with the level of support and stature that this university and the Baker Fellows Program brings. The program supports the research with discretionary funding to help get through what we call the valley of death. So projects that are maybe not fundamental enough anymore to get funding from NIH, and they're definitely not ready for a pitch to industry because they're still so nascent. And at this point, we have over 40 fellows that are part of this community, which is another tremendous aspect of this program. It brings together faculty so that we can talk about our research and also talk about the efforts that are oftentimes being led by our graduate students or postdocs and alumni in terms of looking at how can we commercialize these approaches and make them available to people who need these solutions as products and not through one-off collaborations with our research groups. So this has been a tremendous asset, I think, for the university and something that really distinguishes us from a lot of our peers. Two quick things I'll just mention on this. So Professor Misha Rappe started a company a couple of years ago, Nerex, with some of his colleagues from Berkeley. He is a Baker Fellow and ended up being named to top 50 startups by Forbes magazine, as well as raising substantial levels of funding for that company through 3 rounds of funding and employs over 100 employees right now. In addition to that, Professor James Hurley in May of this year, so just about maybe 2 months ago, co-founded a startup, and that startup was funded at Series A from Third Rock Ventures to the tune of $60 million on a first round. Both of those companies are working on new ways to treat disease. And so without the Baker support and without campus's support, I would agree with Professor Hurley and say some of these efforts wouldn't see this accelerated light of day in terms of commercialization and availability to the patients who need them. So I just add here at the bottom what Jim told me. He essentially said that without the Baker Fellow support, which is UC Berkeley support, he really would not have seen this happen at all, which is an amazing testament to the vision of Berkeley. So the last thing I'll say is therapeutics are one area where we're having an impact. Another is definitely in the biomedical hardware space. And one just last little highlight that I want to give here is a product and a company that's been named as a best product by Time Magazine. The founders were named to Forbes 30 under 30. In addition to that, they are the youngest team of founders we know to have an FDA-approved product, and they actually have 2 now as well. And you can draw a direct line from that effort, this electronic stethoscope, where actually the secret sauce is in the algorithm that decodes the signals that come from that electronic stethoscope. But you could draw a straight line back to the research environment, the resources, and the fellowships that I described a little bit earlier. But actually, I would be a little bit remiss if I left the story there, because it turns out this startup and these products that I just mentioned to you, this electronic stethoscope, didn't happen in a research lab at all. It happened in something that looked a little bit like this, an undergraduate course. So from a lecture hall, something like this team of students came up with this idea. They pursued it as part of their senior project and led to commercialization. And actually, I'm being a little bit too superficial there as well, because most of the training that we do at UC Berkeley, especially for upper division undergraduates, doesn't happen in a lecture hall like this. It happens somewhere more like this. So most of the teaching that I do every day is actually through apprenticeships. So it's through working with my graduate students and postdocs one-on-one or in small groups. And at the undergraduate level, it is bringing graduate students, these leaders of tomorrow, to work directly with undergraduates and faculty like myself around design studios like this. And in fact, this stethoscope that I just described to you also involved clinicians from UC San Francisco. Each of our senior bioengineering teams works directly in a medical environment as part of their senior project. So if you you have a conception of UC Berkeley being very much lecture-based, that is probably true for some of the lower level, the starter fundamental courses, but really, quite honestly, most of what we do is face-to-face interaction around real-world problems and applying those fundamentals that we've learned. The very last thing that I'll say is what is especially important to me is the fact that UC Berkeley, as you probably know, is considered among the top 10 universities doing the most for the American dream. So all of these statistics that I've told you about our standing in the world regarding scholarship and inquiry and education, as well as the numbers that I gave you around entrepreneurship, including for women leading efforts as well, all tie back to this. And that, for me, means that Berkeley is a portal to a world where students really can dream it and they really can do it. And we are here to support them, and you are also supporting them as well. So if it's Connor's stethoscope or it's the tiny molecular stethoscope that I talked about earlier, our goal is to get these ideas and discoveries out into the world through knowledge, so publications and dissertations, but also in terms of getting those tools out into the hands of people who need them through collaborations, and sometimes that also means commercialization as well. And our overall goal is to have a positive impact on the world through both the projects and the research that we undertake, but I think most importantly, through the people that we train and support. Thank you.
So, thank you, Professor Herr. So we have a few minutes. If there's a few questions for Professor Herr, we have a few minutes. Does anybody have a question? You raise your hand, and there's a microphone. We want to hear it. It's very difficult with this light. I don't know how you've done it this whole time. Okay, there's a question up here.
Thank you. And if you could say your name, that would be very helpful.
My name is Linda. And I actually worked in the lab at Stanford that did the technique work that allowed the Human Genome Project to continue. So I understand the importance of your technique, but the professor that I was working with actually was more interested in what he was trying to discover and work on. And you didn't tell me much about what your more, if I can say it this way, scientific goal is rather than your mechanistic goal. You understand what I'm saying?
Yeah, absolutely. I'll try to make it quick. I am an engineer, so I'm more on the applied spectrum than a fundamental scientist, and I'm quite proud of that. I'm a practical person, so it fits well with my personality. A big thing that we're interested in are measuring proteoforms, so isoforms of different proteins that are different forms of proteins and that are out there that are not measurable with existing technologies. So for us, part of our questions are, how many proteoforms are there? Something that can't be measured for a lot of targets right now, and what does that mean? When do they matter and when do they not matter? So I could talk to you more about that fundamental, but that is a discovery aspect of our work. Thank you.
Thank you.
Anyone else?
How is the success in the engineering school in getting more women enrolled, particularly at an undergraduate level? Because nationwide, there's been a big problem getting women into engineering schools. Could you discuss that a little bit?
Yeah. Thank you.
Paul, yeah.
Thank you, Paul. Thank you very much. I think a big part of it is us stressing the amazing aspects of this job. So every day I work on puzzles. Every day I try to figure out how to work with my team and make solutions and discoveries that are not, we don't even know if we can do it. It's a big challenge, and that's something that a lot of people really like. I think another aspect of what we can do is to really highlight how much engineers literally create the world and the future of that world. And being able to play a part in both the creativity and the rigor of making the world what we see it as it should be is something that is just ignites a fire of interest in a lot of people. I think another aspect that could be highlighted is the ability to work with people all over the world. So the collaborative teams that I work with expose me to different cultures and different approaches all the time. So someone who's interested in understanding and learning about different cultures, it's another great avenue. Maybe the last thing I would say that's something that is important to stress, it certainly was for me, coming from a family that was not well off, is my ability to work on really hard challenges that are really fun, but to also be able to generate a salary that can support myself and my family, so making me independent in terms of being able to function in the world. Those are maybe just 4 things I would say that we should definitely highlight. I would also say you do not have to be a genius at math in order to be a fantastic engineer. And I love math, but I didn't always, and you don't have to be a genius. You have to learn what you need to know to be able to solve the problems, and when you have a real problem in front of you, I can guarantee you a lot of women and girls will want to learn exactly what they need to know to solve that problem. So I appreciate your question very much. Thank you.
My name is Richard Hodges. I was a graduate student in math here.
Oh, great.
My question is, have you ever faced and how did you approach the ethical questions of deploying new technologies that have the potential for transforming life at a fundamental level and also the potential for abuse and weaponization and all of these questions that are very pressing these days?
Absolutely. So even understanding the intended and maybe the unintended consequences or uses and taking some responsibility for those at a very early stage. I think, from the perspective of a faculty member, I am a faculty member in STEM, and we're maybe not so well known for paying attention to the social sciences and some of these questions. But I will say, after starting at Berkeley, as I mentioned, almost 11 years ago, I have noticed a couple of things about the culture of this campus that I also tried to expand in my broader technical field, and that is a very big commitment to not only making an impact through our studies and our research, but making sure that that is a positive impact from the research and the studies that we undertake here. And I think part of that is, from the very early days in the engineering curriculum, being required to take courses in bioethics, not stale courses, but courses that are talking about exciting technologies like CRISPR, for example, and thinking about what are the possibilities with curing diseases we couldn't even touch maybe 5 years ago, but also the flip side, what could go wrong with this in terms of unintended consequences or malicious intended consequences, and having very serious upfront discussions about that. Certainly a big area that I teach is engineering design. One of the biggest steps of the engineering design process is the last step, which is reflection, which is standing back and thinking about, have I asked all of the questions I should be asking? Have I lived up to my responsibility as an engineer? And what do I need to do if my answer to those questions is no? So I very much appreciate your comment. Thank you.
If we have time for one more, if there is one more.
Yeah, and I have a question here on a card that I'll ask for one of our audience members. So, Amy, can you please give an example of how a genome or DNA analysis gave a diagnosis for successful treatment of cancer or another serious disease, which regular medical diagnostics may have missed?
Yeah, so there's actually a whole set of them that we could talk about and certainly give an entire talk on, but definitely looking for any of these mutations that may manifest in terms of types of very specific or aggressive cancer, for example. I think maybe many people here are familiar with the BRCA gene, for example. So being able to determine early when your body may be sort of predisposed, if you will, to developing an aggressive form of breast cancer even before it would be detectable, using that information even before it would be detectable with, say, mammography. I think if I might just dovetail from that for a moment into my own field, there's a lot of questions like that that are specific also to proteins. So we know the Human Genome Project. Many of us are interested in the Human Proteome Project, which is really thinking about how does disease manifest when bad stuff happens to these millions of proteins that are inside of our body. A big area that we're working on is HER2 positive breast cancer. The reason that we're working on that is because some of the amazing therapies that have been developed for HER2-positive breast cancer are completely ineffective when the HER2 protein is missing a part of that protein. It gets truncated. And that's one of the mechanisms that disease uses to circumvent treatment, response to treatment. It basically outsmarts us in some way. So for us, we're pretty interested in these dynamic changes at protein level, but certainly the example that I just gave and many, many other tie back to the genome as well.
Please join me again in thanking Professor Herr.